So hello everybody. We'll start with our regular announcement. We know that the research day is approaching. We receive over 70 abstracts. So what is left is to reserve a spot. So please go there and book your place. As you know, thank you for keeping your camera active during the seminar. Last week, we talked about Fem Therapeutics, who offers 3 internships position in the summer. We have left only one through the medical technology internship 1 and 2. I'm going to tell you a bit about this position. So if you're interested, please apply to our course for the summer. So they are looking for a motivated intern, 3 months, eager to learn, grow, and build clinician patient-focused tech. Requirements: analytical individuals with passion for working in the field of medicine, strong biomedical, data science, software, or mechanical background, good understanding of data handling, analysis, and programming on structured and unstructured data. Thank you very much. Now, Dr. Moale will present our lecture.
Yeah, so just a few words about Fem Therapeutics. So these are students who, former students who are taking our course, Surgical Innovation, and they started a company, and the company is just near here in Montreal. And recently they got $4 million, and before they got about $2 million. So really successful team. If you wanted to do an internship, this would be the right time to do with such a wonderful homegrown team. So it gives me immense pleasure to introduce our distinguished speaker for today, Dr. Melica Brodeur. She serves as an assistant professor in the Department of Obstetrics and Gynecology and Oncology at McGill, a testament to her expertise and dedication in her field. So Dr. Brodeur is not only an attending gynecologic oncologist, but also a principal investigator at the Jewish General Hospital and the Lady Davis Institute for Medical Research. Her dual role as a clinician scientist enables her to bridge the gap between patient care and innovative research, embodying the very essence of translational medicine. Her academic journey began at McGill, where she completed her medical degree. Driven by a passion for obstetrics and gynecology, she then completed her residence at the University of British Columbia, followed by a subspecialty in gynecologic oncology at the Université de Montréal. So her quest for knowledge didn't stop there. She further honed her research skills with a master's in biomedical sciences and later pursued a postdoc research fellowship at the prestigious Memorial Sloan Kettering Cancer Center. Dr. Brodeur's research is important in understanding how gynecologic cancers respond to treatments, striving to unveil new therapeutic targets and strategies to combat therapy failure. She co-leads the Gynecologic Oncology Research Lab, where her work has not only advanced our knowledge, but has also earned the Ovarian Cancer Research Alliance Early Career Investigator Award. But beyond her impressive credentials and contributions to science, what truly stands out is Dr. Brodeur's mission to improve the lives of patients and families affected by gynecologic cancers. Today, she brings to us insights from her research and a message of hope for the future of cancer treatment. Please join me in welcoming Dr. Melica Brodeur, and the title of her talk is Modulating Chromatin Remodeling as a Strategy to Reverse Immune Evasion in Ovarian Cancer. Dr. Brodeur.
Hi, thank you so much for the introduction. I am really quite honored to have been invited to speak at this seminar series. I understand that the audience come from different backgrounds, and so hopefully parts of the talk will tie into some of your interests. And please feel free to ask any questions. You can put them in the chat and we can review them at the end of the talk. So as you think of them, please go ahead and add the questions. And if there's any problems at some point, just interrupt me, let me know. And hopefully you'll have learned something by the end of this talk. So indeed, the title of my talk is Modulating Chromatin Remodeling as a Strategy to Reverse Immune Evasion in the Ovarian Tumor Microenvironment. So given everyone's different backgrounds, I thought I'd talk a little bit about ovarian cancer as a whole. So you may or may not be familiar with the fact that ovarian cancer is now it's now known that it's not a single disease entity. There are different types, with the most common type being epithelial ovarian cancer, and it's divided into 5 main subtypes listed here. And then there are the non-epithelial ovarian cancers that I kind of put in this black box because we kind of throw them in this sort of other category. And that can include ovarian cancers of germ cell type, sex cord type, and also SCOOTS, which stand for small cell carcinoma of the ovary hypercalcemic type, which is a type of rhabdoid tumor. And unfortunately, if you've heard of ovarian cancer, you've probably heard the cancer statistics that show that ovarian cancer is still a deadly disease. Most patients present in advanced stages, and despite the known heterogeneity of the disease, we're giving patients all the same first-line cytotoxic treatment rather than personalizing the treatment approach for the different types of ovarian cancer. And so unfortunately, the 5-year survival remains quite low at 30%, given the late presentation of these patients. And so we definitely need to do better for our ovarian cancer patients. You may have heard of immune-targeted therapies, such as immune checkpoint blockade. This group of therapies have shown dramatic success in multiple cancer types, including some gynecologic cancers like endometrial cancer and cervical cancer. But unfortunately, until now, immune checkpoint blockade trials in patients with ovarian cancers have shown really disappointing response rates with no clear biomarker for response. And so despite that, these studies have revealed when sub-analysis have been performed that a subset of ovarian cancers called clear cell ovarian cancers have a higher response rate, and it may be clearer towards the end of the talk why that might be. Shifting gears, epigenetic therapies, including things like targeting chromatin regulatory factors or their downstream pathways, have emerged as promising therapeutic strategies in recent years. And so the SWI/SNF complex plays an important role in gene regulation. It's a chromatin remodeling complex, and it has roles in regulating oncogenes, tumor suppressor genes, and I usually like to compare the SWI/SNF complex to like an on-off switch of gene transcription, put sort of very briefly. The complex is made of multiple subunits, and 29 genes encode for these different components. And here in the box, SMARCA2 and SMARCA4 are 2 critical ATPase subunits that utilize energy from ATP hydrolysis to mobilize nucleosomes here depicted and remodel chromatin. So essentially making chromatin more or less accessible to transcription. And so the SMARCA4 subunit here, whose protein is also known as BRG1, maybe that's a term that you're more familiar with, will be what I highlight throughout this talk. So several studies demonstrated that a subset of ovarian cancers are characterized by a more immunogenic tumor microenvironment that could make them potentially more sensitive to immunotherapy. And so before I joined the group at Sloan Kettering, the research team had characterized the immune tumor microenvironment of a series of patients with SMARCA4 mutated tumors, particularly SCOOTS, so the small cell carcinoma of the ovary hypercalcemic type of ovarian cancer. And in this study, it was found that there was a prominent T cell infiltration, here depicted by the marker CD3 positive in the tumor microenvironment, and also tumor-associated macrophages that were highly expressing PD-L1 here. And so also mentioned in the paper, anecdotally, some of these patients responded to immunotherapy, whereas they hadn't responded at all to standard of care chemotherapy. And so this is interesting in the context of these tumors that are known to have quite low tumor mutational burden. And if you know anything about what tumor mutational burden is, it typically has been associated as a biomarker for more immunogenic tumors. This was quite an interesting finding. We then actually queried the TCGA data, and what we found when we looked at high-expressing SMARCA4 ovarian cancers versus low-expressing SMARCA4 ovarian cancers, we found that the low-expressing SMARCA4 ovarian cancers had upregulated pro-inflammatory immune cell population infiltration. So we have examples here of increased leukocytes, Th1 cells, which are pro-inflammatory cells, and M1 macrophages, which are pro-inflammatory macrophages. And so with this in mind, we wanted to understand the immune tumor microenvironment interactions and mechanisms of increased immunogenicity of SMARCA4 altered ovarian cancers that could imply a potential role for targeted therapy in ovarian cancer. So of course, given that there aren't a whole lot of preclinical models in ovarian cancer, we did have to develop SMARCA4 deficient human and murine ovarian cancer models with their respective controls, of course, in order to study this research question. So we used 2 murine ovarian cancer models. So you have your ID8, which is a TP53 wild type murine ovarian cancer, and UPK10, which is a TP53 mutated murine ovarian cancer. And given that we would be conducting immune tumor microenvironment studies, both our in vitro and our in vivo studies were primarily focused with these murine ovarian cancer models, with, of course, some in vitro validation using a human ovarian cancer model called OAW28, which happens to also be TP53 mutated. And so with the murine models, we derived clonal cell lines that were transfected either with single guide RNA targeting SMARCA4 or non-target control using the CRISPR-Cas9 technology. And so once our models were ready, and that did take a few months, we performed RNA sequencing, comparing the expression changes with or without SMARCA4 deficiency. And what we found was that downstream immune-related genes and pathways were upregulated, including important interferon-stimulated genes, chemokines, as well as antigen presentation genes. And you can see that as well with the enriched pathways that were found in these SMARCA4 knockout cells. You see your interferon as well as antigen presentation pathways. So with this data, we focused on these specific downstream immune pathways, and in particular, the type 1 interferon genes were significantly upregulated. As you can see here on the left panel, these are lists of genes that are part of the type 1 interferon pathway. And so you could see here that the knockout cells were, generally speaking, more upregulated compared to the non-target control here on the left. And we were able to further support our RNA-seq analysis with our flow cytometry data, where we found here on the right panel a significant increase in MHC1 expression on the surface of our SMARCA4 knockout cells compared to control, which suggested an increased antigen presentation ability of tumor cells with loss of function of SMARCA4. And so here you can see in black the SMARCA4 wild type cells, and then in blue here, the SMARCA4 knockout cells, and then the same here with the addition of interferon gamma, which tends to sort of mimic an activated tumor microenvironment in vitro. And so given SMARCA4's role in the chromatin remodeling complex, we next wanted to understand whether loss of SMARCA4 led to changes in chromatin accessibility that could explain some of the immune-related changes that we were observing. And so we performed attack sequencing, and amongst the genes that demonstrated increased chromatin accessibility, interferon and chemokine signaling pathways were quite upregulated. And many of the genes that had increased expression in our RNA-seq analysis also had increased accessibility on ATAC sequencing. And so I gave here two examples, so CCL2 and CXCL10. These are downstream chemokines of the type 1 interferon pathway. And in red here, you have the SMARCA4 knockout cells compared to in black, the wild type cells. And you can see here that there's a peak at the promoter site compared to the non-target control peak here in blue. And so we then further analyzed the data to determine if there were any recurring motifs that were enriched in the open chromosomal regions affected by SMARCA4 deficiency. And we identified at least 3 significantly enriched motifs that are bound by transcription factors that regulate immune genes, including AP1, NF-kappa beta, and interferon regulatory factor, which I've boxed out here as we're going to be discussing this in particular. So with these data so far, we wanted to further explore the interferon-stimulated gene upregulation mediated by the type 1 interferon signaling pathway. So for those of you that may or may not be really familiar with this pathway, ISGs, which is interferon-stimulated genes, their transcription is initiated when type 1 interferon receptor is stimulated by the secretion of interferon alpha and beta. And interferon alpha and beta are produced in the nucleus after key upstream regulators such as IRFs here. So you have IRF3, IRF7, IRF5 that get activated by a stimulus, which could be from upstream triggering events like DNA damage or increased double-strand RNA or non-coding retro elements. And so our aim was to understand and test the key players in this pathway that led to the interferon-stimulated gene induction. So we started with blocking the type 1 interferon pathway, the type 1 interferon receptor here. However, what was interesting was that when we performed this neutralizing assay to block the receptor, we found that it actually did not attenuate the interferon-stimulated gene expression in our SMARCA4 knockout cells. So on the left panel here, you see that the SMARCA4 wild-type cells are in black and gray, and the colored bars are the different SMARCA4 knockout clonal cell lines treated with or without the receptor blocker. And so if you take, for example, SMARCA4 knockout cell line 4 here in purple, you can see with and without treatment, there's no significant abrogation of the interferon-stimulated gene upregulation. And so these suggest that transcription activation of these ISGs are not fully dependent on activation of the type 1 interferon receptor by secreted interferon alpha or beta. And so given these data, we then proceeded to evaluate whether key upstream regulators, as I mentioned before, such as IRFs, were directly responsible for activating the ISG, kind of bypassing this interferon alpha receptor. And so what we found is that both IRF3 expression and protein levels, here with the Western blot below, were upregulated in SMARCA4 knockout cells compared to control. So here you have the colored SMARCA4 knockout clonal cell lines compared to the wild type cells, and you can see here that there's significant upregulation at the expression at the gene level as well as the protein level. And not only was the total expression increased, but also the activated IRF3 or phosphorylated IRF3 that is measured by nuclear to cytoplasmic ratio of IRF3 in the context of loss of function of SMARCA4. You can see here that SMARCA4 knockout compared to the control has significant increased nuclear presence of IRF3. And here in gray is the positive control. So then to be able to actually link IRF3 induction with ISG upregulation, we performed knockdown experiments using shRNA targeting IRF3 or of course non-target control in the SMARCA4 knockout cells. So what we saw was that knocking down IRF3 abrogated the ISG upregulation, reinforcing the direct link between IRF and ISGs. So in the panel here, the black bar is the SMARCA4 wild-type cells, and in the blue and orange are the SMARCA4 knockout cells transfected with either SH non-target control in blue, or in orange is the SH-IRF3. And you can see here that the upregulated ISGs, which are these at the bottom here listed, are increased in the SMARCA4 knockout cells and get abrogated with the SH-IRF3. So with these findings, we decided to examine the IRF3 binding sites at the ISG promoters as a potential mechanism that could explain the interferon alpha receptor independent ISG upregulation that we observed with SMARCA4 loss. And so using our attack-seq data, we found that IRF3 motifs are enriched at the transcription start site of ISG loci. And I gave here an example of an ISG, which is OSL2. And this suggests that IRF3 may bind some sort of interferon-stimulated response element that directly triggers ISG expression. So you can see here in red is the SMARCA4 knockout cells, and the peak is quite higher at this transcription site, start site, compared to the wild type cells. And we also explored publicly available data sets from ChIP-Atlas showing IRF3 binding sites at the promoters of key ISGs. So again, I used OASL as an example in human cancer cell lines. So here I put the example of the MM1S cancer cell line. You can see here that there's an IRF3 binding site at the transcription start site of this gene. And we also see the same in mirroring immune cell lines. So here again, I used OASL2 as an example of an ISG, and you can see here that there's IRF binding sites present at the promoter of these of this gene. So knowing that interferon-stimulated gene upregulation was not dependent on type 1 interferon receptor and seemed to be directly affected by IRF3 activation, we focused on potential upstream signaling mechanisms leading to IRF3 activation. So I kind of alluded to that earlier. That can be from DNA damage or non-coding retroelements or increased double-strand RNAs. And so you can see here, this is where IRF3 is, and we decided to focus actually on the RNA sensing pathway based on some literature that previously suggested that this could be an important factor in triggering the type 1 interferon pathway. We hypothesized that chromatin remodeling associated with the loss of SMARCA4 could lead to re-expression of non-coding or transposable elements, which carry a potential to engage innate receptors that have been shown to lead to type 1 interferon pathway activation in other settings, for example, in viral infection. And we've also seen these types of results with inhibition of DNMT, for example. So we focused on the DNA sensing, the RNA sensing pathway, and the three main sensors are listed here, which is RIG-I, MDA5, and MAVS. So what we did is we first analyzed our RNA-seq data looking at non-coding genes, as I mentioned, and what we found is that actually loss of function of SMARCA4 in our murine ID8 cells induced a significant increase in a type of double-strand RNA or transposable element called long terminal repeats. And this category of transposable elements includes endogenous retroviral genes, which you may or may not have heard of, and has gained a lot of traction recently in this field of research. And what we did is once we saw this data, we wanted to confirm it in a second model. So we used our UPK10 model, and we were with a panel of endogenous retroviral genes. We were able to also see that there was quite a significant upregulation of these non-coding elements in cell lines that had lost SMARCA4 function. So with these findings, we wanted to determine whether the RNA sensing pathway could potentially be driving the ISG upregulation, possibly through detecting increased long-term no repeats expression in SMARCA4 knockout cells. So to confirm this association, we performed knockdown experiments. Specifically, we knocked down MAVS, which stands for mitochondrial antiviral signaling protein. And it is a key downstream adapter and RNA sensor that mediates both RIG-I and MDA5 signaling. So what we saw was that the knockdown of MAVS versus the non-target control in our SMARCA4 knockout cells resulted in downregulation of expression of different ISGs. So you can see here on the right panel in black, we have the SMARCA4 wild-type cells. In orange, we have the SMARCA4 knockout cells that are transfected with the non-target control. And in green and blue, we have the SMARCA4 knockout cells that were transfected with an SH MAVS. And you can see here the abrogation, generally speaking, across ISGs when MAVS is knocked down. So with these in vitro data, we wanted to see if this increase in immunogenicity was observed in the tumor microenvironment. Because, of course, it's all great to see all these in vitro experiments that were very promising, but if we don't actually, if we're not able to actually recapitulate these results in vivo, they're sort of futile. And so what we did is we inoculated immunocompetent mice intraperitoneally with SMARCA4 knockout versus non-target control cells. And we collected the ascites of these tumors after 3 weeks. We then performed spectral flow cytometry to examine a panel of innate and adaptive immune markers. And I can tell you that we got many results, but I've highlighted some important ones for you. And so in a nutshell, what we noticed was that our CD4 and CD8 positive T cells were significantly more activated in our SMARCA4 knockout cells, here depicted in blue compared to the control in black. And this was seen by the increase in PD-1 expression in both CD4 and CD8 cells. Our natural killer cells were also significantly more active, as seen by the increase in granzyme B expression in our natural killer cells. We also had a look at the myeloid compartment, and so we also, saw a lot of interesting changes in the tumor microenvironment as well. And so firstly, there was a significant increase in dendritic cells, and these dendritic cells highly express MHC2, suggesting that they were more capable of antigen presentation. And then in parallel, we also imaged our mice, and what we were able to notice was that the tumor velocity was significantly impaired in our SMARCA4 knockout tumors, which suggested that there was potentially some sort of immune anti-tumor suppressive effect of SMARCA4 loss of function. So we next wanted to see if we could therapeutically target SMARCA4 and recapitulate these results. And so what we did was that we inoculated our mice with ID8 cells, but this time it was just the parental cells. And we treated the mice with commercially available SMARCA4 inhibitor, which is called the BRG1 inhibitor, or vehicle. And as you can see on the right panel, the CD4 and CD8 cells were significantly more activated. And you can see that by the increase in ICOS and PD-1 expression in our CD4 cells here. And at the bottom, you can appreciate that with the increase in ICOS, PD-1, and granzyme B expression in our CD8 positive T cells. We also looked at the myeloid compartment as well. And what we found was, again, that the dendritic cells were more prominent in the tumor microenvironment. And they also appeared significantly more antigen presenting given the increase in MHC2 expression that we found. In parallel, we also looked at growth velocity of the tumors with and without treatment, and we found that the treated tumors also had a suppressive growth effect by both imaging as well as weight measurements for ascites accumulation. So with these results, we were able to conclude that the disruption of the catalytic subunit of the SWI/SNF complex led to increased tumor immunogenicity. That type 1 this increased immunogenicity was type 1 interferon receptor independent, and actually likely more mediated through interferon regulatory factors. And it may potentially prime the tumor microenvironment to be sensitized to immune-targeted therapies when there's a loss of function of SMARCA4. And so at the moment, we're conducting some studies to add immune checkpoint blockade to see if we can sensitize these tumors to respond to this type of treatment. And we're also exploring combining BRG1 inhibitor with other epitherapies such as HDAC inhibitors or DNMT inhibitors, which have also shown to be able to activate the type 1 interferon pathway. And so with that, I'd like to acknowledge important people that have helped with this work. And so namely members in my lab. So Olivia McKeeman, she's a master's student that is pursuing this work here at the Lady Davis Institute. Amber, Yasmin, and Marwa Mubarak are research associates in the lab that have tremendously helped on this project, as well as Sam, our research technician. And the core facilities at the LDI, which have also helped significantly with developing protocols to make this project possible. And of course, my mentors during my postdoc at MSK, both Dr. Weigelt and Zamarin, and the cores at Memorial Sloan Kettering that also helped this work be possible, and other mentors that have contributed significantly to the project, including Kate Chapinelli, who's in Washington, D.C. And of course, none of this work is possible without funds. And so I'd like to also give a shout out to the FRCSC and OCRA, as well as FRCSC, that have tremendously supported this work, without which it would not be possible to explore this research path. So I'd be happy to take any questions. I've left my email here if ever anyone wants to get in touch or has any thoughts about this project. I'd be more than happy to discuss further. So yeah, thank you so much for your attention.
So thank you very much, Professor Brodeur, for that wonderful presentation. Any questions?
Sure, I have one. Thank you, Dr. Brodeur. That was a wonderful presentation, very thorough. I really, really liked it and enjoyed all the results that you had to show, especially with the RNA-seq and at the end with the inhibitors and showing how that improved, I guess, the immunogenicity in your cancer models. I just had a question related because so you did knock down for this SMARCA4 to show increased a little bit immunogenicity, I guess, or that's related in the cancer in ovarian cancer models. And I think in some tumors, or at least there's, I mean, correct me if I'm wrong, but in some ongoing tumor models, increased immunogenicity could also lead to tumorigenicity is one thing. So I was wondering, have you looked in the opposite? Have you actually upregulated, SMARCA4, and seen if there was a different effect or an enhanced, let's say, carcinogenic effect with the in oncologic models?
So thanks, Michael. That's a great question. I actually I kind of got validation for this work through it was towards the end of my postdoc. I attended a lecture by Dr. Hargreaves, who also works in this space. And as I was attending her lecture, she had basically done almost the same project as I, but with ARID1A. And so she knocked out ARID1A in her models, and I could literally, the slides that she was showing me was basically my slides. And so for a moment I panicked, but then I actually realized this is great because it validates our work and actually tells us that probably the downstream effects that we're seeing on the type 1 interferon pathway may not necessarily be specific to SMARCA4 or ARID1A, rather that it's a disruption of the Swiss-Steve complex that leads to these findings. And so just based on her work, I'm actually waiting for her to publish the work. But based on her findings, it actually validated the work that we had done, supporting, in our case at least, that the loss of function of important subunits of the Swiss-Steve complex actually led to more increased immunogenicity and an immune anti-tumor effect, because she also had imaged her mice and found that there was this suppressive effect on ARID1A knockout tumors. And so I felt more confident with our results after attending her lecture. So hopefully that sort of indirectly answers your question. I don't know if it's been formally studied to see if in any way, shape, or form it can increase tumorigenesis, but at least so far it seems like it doesn't.
Sounds good. No, thank you.
Welcome.
Any other questions? Yes, this is very elegant work and really well done. So in terms of, I think I'll piggyback on Mike's question in terms of its relation to SMARC4, but about the therapeutic vulnerabilities, given the increased immunogenicity which is associated with this SMARC4 loss, what do you think are the potential vulnerabilities that are exposed in this SMARC4 mutated ovarian cancers? Can you be able to target this with some sort of therapies?
Well, honestly, that's what we're hoping. Beyond these type of epitherapies, because BRG1 inhibitors, because it's an inhibitor of a subunit of the Swiss-Steve complex, it still falls under the epigenetic therapies. And so I think that epigenetic therapy as monotherapy has not shown tremendous success in clinical trials. And so I definitely think that for sure combination therapy is going to be necessary. Now, is the answer, 2 epitherapies? I'm not convinced, unless they have significant sort of different mechanisms of action beyond just activating the type 1 interferon pathway. And so that's why I think that rather using these epigenetic therapies as an immunomodulator. So not so much as an anti-tumor, having an anti-tumor effect directly, but rather immunomodulating the tumor microenvironment to then respond to immune checkpoint blockade, which to date in ovarian cancer has had really mediocre results. And so this is likely from a very immunosuppressed tumor microenvironment. And so if we can sort of reverse that, maybe we'll have more success, as we've seen with other gynecologic cancers and other sort of cancer types like melanoma, lung cancer, et cetera. So that's sort of the hope and the rationale for now. So maybe in a few years I can let you know.
Yeah, I was also intrigued about the knockdown of these RNA sensors. That was quite well done. So what do you think is happening?
Yeah, well, I think, to be honest, when the Swiss-Steve complex is disrupted, I think it actually has a tremendous amount of effects. I mean, we've focused on, sort of one part of the effects that it might have, but I think that the disruption is such that there are now re-expression of non-coding elements that, to date, people haven't really paid much attention to. And I think it's only been in the last few years, maybe in the last 10 years, which, maybe, 5-10 years, where we've started to realize that these non-coding genes may actually hold some importance in terms of being able to activate these type of pathways. And I think that's probably what's happening. There's maybe the re-expression is occurring because of previously methylated genes that are now being demethylated once the Sweet-SNP complex is completely disrupted, and allowing, the chromatin accessibility in places that normally it wouldn't be accessible. So I think that's probably what's happening. And of course, you'd have to do lots of studies to be able to actually show that correlation. But that's what I suspect, based on other people's work that have investigated more formally this re-expression phenomenon of non-coding genes.
Nice. Any other questions? Just unmute.
I'm going to take that it was very clear.
Yeah.
It's a lot to absorb. It's a very epitherapies is epigenetics, I would say, is a fairly new branch of research, and so it's not known to everyone. But hopefully, I've been able to sort of teach you or enlighten you on this field of research.
Yeah.
One more part, just a quick question. So related to at-risk patients, so anybody who's at risk, preventative measures, preventative therapies. So if I know oxidative stress, you hear a lot, plays a role in a lot of cancers in the formation of, but once developed, I guess inhibiting oxidative stress could have sometimes negative effects. So in this particular, for ovarian cancer, is there any preventative measures that can be taken for those patients who are at risk of developing?
It's actually really interesting that you mentioned this. So as I had alluded to at the beginning, ovarian cancer, is really not one disease entity, and the pathogenesis for the different subtypes of ovarian cancer really differs. So the most common epithelial ovarian cancer is high-grade serous ovarian cancer, and so there are patients with, for example, hereditary syndromes like BRCA mutations that put them more at risk. But the pathogenesis to high-grade serous ovarian cancer is very unique and not so much driven by oxidative stress. However, something that I learned fairly recently in putting together a grant is that the clear cell ovarian cancers and the endometrioid ovarian cancers, which we think stem at least in part by the endometriosis disease, we think that in these endometriosis cysts, there is quite a lot of upregulation of oxidative stress pathways, which eventually leads to carcinogenesis and the development of clear cell and endometrioid type of ovarian cancer from these endometriosis cysts, which endometriosis is by definition benign as a disease. And so potentially in certain subtypes of ovarian cancer, those are pathways that where maybe if patients are known to have endometriosis, could one target in a preventative manner with these epitherapies? Possibly. It's an interesting thought. Yeah. Thank you so much for the questions.
Oh, thanks. Thanks for the answer.
So there are no biomarkers to detect early ovarian cancer. Are there?
No, unfortunately. And I think whoever will discover that will have a Nobel Prize of Medicine. It's been large trials. So there's screening, and then there's early diagnosis. So screening has been looked at in 2 large, very expensive trials that were conducted. One in the UK that very clearly showed that the current strategies to screen ovarian cancer do not work. So that includes things like ultrasound, serum biochemical markers, and in terms of early diagnosis, we're just not able to, because by the time patients have symptoms, these symptoms are related to advanced stage disease. And so there is a lot of work, and grants have been targeting the field of early diagnosis and screening in ovarian cancer, because this is where we could really make a change. Because once it's diagnosed late, we don't really have good treatments to be able to cure these patients. You know, we have more palliative type of treatments to prolong life, but not so much for cure. And so I feel like both spaces, the therapeutic side is really trying to be developed, as well as the early detection and, you know, biomarker screening. So there's still a lot of work to do, but for the longest time, gynecologic cancer research was very underfunded. And so it's just recently, really, like in the last 5 years, where funding is starting to increase in this space. So it'll take some time before we see the results of that money that's being put into gynecologic cancer research. So let's speak in about 5-10 years' time.
Thank you very much for that wonderful, stimulating presentation. And thank you everyone for attending, and have a wonderful, have a wonderful vacation starting from Friday, Good Friday. And enjoy your turkeys. Thank you very much.
Thanks, everyone. Bye.
Bye, everyone.
Bye.